Hi, my name is Larsson Omberg. I am a researcher at Koneksa Health. And in recognition of Parkinson's Awareness Month, I would like to talk a little bit about what Koneksa does to better understand Parkinson's disease. So Parkinson's disease is a very complicated disease and very, very varied. The Michael J. Fox Foundation likes to use an adage that says that if you met one Parkinson's patient, you've met just one Parkinson's patient. And part of this is because how Parkinson's disease is experienced from a patient perspective can be very varied. One of the hallmarks of Parkinson's disease is tremors, for example, but only about 70% of patients actually experience tremor throughout their Parkinson's disease. Likewise, what patients really care about often differs somewhat what clinicians are willing to measure in the clinic or what they're able to measure in the clinic. And with Koneksa's mission to focus on contributing to better lives by building health measures that matter, this is very important to us, to make sure that we're measuring things that actually matters to patients. While we serve primarily the clinical trials industry and trying to advance the development of new drugs in Parkinson's disease, we also know that what needs to be measured in those should matter to patients. And one of the things is, in the traditional sense of determining how your disease is or how Parkinson's patient's disease is progressing, is you might go in and see your clinician once every 6 months or 12 months or so, and you get a very sporadic assessment of it. Well, the approach that we have taken at Koneksa is to consider basically the measurement of the disease in your home using things such as a smart device or a wearable device that could basically monitor the disease throughout the lived experience. So what I'm showing behind me right here is an example of a video of one of the assessments that we use in the home that takes about a minute or so, and it uses basically finger tapping. So this might be familiar to what a clinician might do, observing basically how a patient is tapping like this. Instead, we're using the smart device to measure the tapping speed, but we're doing things beyond tapping speed. We're also measuring things such as variability in tapping. So you can measure things such as tremor or bradykinesia, which would be slowness. Here's another example of something that we measure. We use a traditional 20-second walk test, which is often used in the clinic as well. But in this case, we're asking a patient, and from the video, that's actually me walking right there, ask the patient to walk for 20 seconds and then put their phone in their pocket as they're walking. And in the phone, there are sensors that we can use to assess basically what's happening in the gait process. These sensors, such as accelerometers and gyroscope, measure both the movement of the phone, both in a linear direction as well as the rotation of the phone using the gyroscope. And what you get out of this is basically what I'm showing on the screen right here is basically a time series of movement of the phone in the X direction, Y direction, and the Z direction. And from these, we at Koneksa have developed algorithms that then extract dynamics for gait, or in the case of tapping, we extract information about the tapping. For gait, these things could be such as the cadence of gait, or the distance that someone walked, or freeze index. Common symptoms for Parkinson's patients is the freezing of gait, so we can measure those kind of things. And then from those, we basically use data analysis, machine learning, statistics, AI to infer things about the patient. So here, for example, I'm plotting in blue the finger tapping test done on a patient that a patient performed over a 6-month period. And what you can see is that patient had a lot of variation in their finger tapping, but there was no progression in the finger tapping. The equivalent here, and this is done for illustrative purposes, is the red dots, which is once every 3 months, the same patient came in and saw their clinician, and the clinician rated them on how fast they tapped and how well they tapped. And from that, you can see that in at the beginning, they had no disturbance in their tapping. But at 3 months, there was mild, and then slightly more at 6 months. And from that, a clinician might assume that there's progression in this. But if you compare that to the variation that's seen at home, you can see that probably the patient just had a bad day or came in at a bad moment of the day when they did these towards the end of the study compared to the beginning of the study. There might be a slight trend in the tapping speed that you see from in-home measurements, and there's maybe a little bit more variation at the end, but very little progression compared to that. And this is one of the advantages of doing frequent in-home assessments, even though they only take about a minute or so. Here's another patient that is further along in their disease. So this is someone who's tapping significantly slower on average, about 60 taps in a 20-second window. And the reason I picked this patient because they're later in their stage, and so they've experienced strong on-off fluctuations. And if you can see that that basically shows up in this tapping speed. So the green dots are tapping that was performed after they're taking their levodopa medication, while the blue ones are where they are before their medication, so closer to their off state. Red dots are ones that were ambiguous, where they were on or off. And so from this, you can see that this patient experiences strong on-off fluctuations, and we can measure those in the home. Likewise, if you plot the same data, but instead of plotting it on a time across months, you plotted a time when they performed the task at home. And you can see that this patient actually has some diurnal variation, meaning that they perform better in the afternoon than they do in the morning. And it turns out that this is independent or on top of the effect of taking the medication as well. And these are the kinds of things that we can discern, that basically what are the effects that are affecting how the Parkinson's disease is showing up in the patient. If we look at this a lot across a large number of patients, we can see that basically, yes, tapping speed is associated with levodopa intake, walking at a slightly lesser effect of this as well. So basically showing here the effect of levodopa before and just after medication. And interestingly enough, if you look at a large population from this, you can see that actually how people respond to their disease is also unique. So not only is it what symptoms people are experiencing are slightly different from each other, but how they're responding to standard of care. And what we are applying this to is really about how can we help measure to better decide if a new drug works or doesn't work. And so we're trying to move away from what's traditionally been used measurements in the clinic, such as the MDS-UPDRS, which is a clinician-assessed measurement, which tends to be subjective. It's expensive and requires patients to travel to the clinic, so it tends to be infrequent and can be kind of imprecise, to something that is more precise, objectively measured using sensors, and then is low burden and can be performed frequently. Thank you very much for spending the time to listen to me.